Okay, welcome back to the second part of our immune response lecture. In this one, we're going to actually start looking at the specific side of the human immune response. Now, because that's quite a complex process, we're going to look at this in 4 stages. So we're going to, I'll put it on the board here. I know my writing isn't the best. But up here, we're going to start by looking at the introduction to the specific immune response and what that is. So all the stuff here is that introduction. Then in the second of these lectures, we're going to look at cell-mediated specific immune response. And then in the third, we'll look at humorally mediated immune response. And finally, we'll look at hypersensitivity responses, because there's 4 classes of them, and it's worthwhile knowing what they are and how they act. So for this one, we're going to look essentially at the whole thing in brief. So we're going to look at what specific immunity is. Now, as the name suggests, there is a recognition factor required to be specific about something. You have to pin down, recognize, and act upon it. So in specific immunity, we have a level of specificity and also of memory. Now, in specificity, this comes through major histocompatibility complexes or classes. Now, in the human situation, we have 2 classes. We have MHC class 1, MHC class 2. In this introduction, we're going to focus on the second one at the end when we look at antigen presenting cells. So specificity is created by these specialist proteins sitting on the outside of the cell being identified by the immune response. And basically MHC class 1 says, I'm one of you, I'm okay. So it's like kind of wearing a football top or something like that. If you're all a Wolverine supporter or a Spartan supporter, you're all wearing your colours, and you walk along the street and you wave at each other as long as you're wearing the right strip. So this is present on all host nucleated cells, meaning cells with nuclei. So that doesn't count for red blood cells because they don't have nuclei in them. But other cells have this MHC class 1 protein sitting on the outside of the cell, and they have proteins sat on them which are from the inside of that cell, and that is recognized by the guards, if you like, the B cells and the T cells. So we also have specificity and memory. It's important to be able to check these for problems and destroy the cell if there is a problem. So that's kind of like a passport or a ticket or something. It's like you have a ticket, you can play. You don't have a ticket, you must leave. Memory is kind of about speed of response. If we go back to the bouncer analogy, you have someone that came to the club the week before, got in, and then caused huge amounts of trouble, damaged a lot of property, etc., and got thrown out. Of course, the bouncers on the door the week later, even if they weren't there the week before, will have been told about this individual and to look out for them. So this is the process of memory. So when a bacteria, for example, gets back into your system and you've already been infected by it, and this is very common in households when we have something like a viral infections like common cold, or even bacterial infections, etc. And so one person comes home ill, and then lo and behold, after 2 or 3 weeks, everybody's had the same illness in the family. But you'll notice that everyone has it once and then you move on. The person that came home ill first gets better, and while everybody else is sick, they don't get sick again. And this is because of the memory that they have here. As soon as they get re-inoculated with the same pathogen, the cells kick in immediately and destroy the invading pathogen, because they know what to look out for. I mean, again, going back to the bouncer analogy, if you're standing at the door and you've got 300 people coming in, and you don't know who's going to cause trouble and who is not going to cause trouble of those 300. If they have the right ticket, you let them in, and you take your chance. But if you have a picture of one person that you cannot let in because they caused trouble before, and that's your memory, as soon as they come to the door, they are stopped, they are removed. They do not get into the cell, or in this case the nightclub, so therefore they can't cause any damage. So we have specificity and we have memory in our specific immune response. Now this is created by major histocompatibility class 1s on our nucleated cells and class 2s found on our antigen presenting cells, which I'll talk about in a minute. It's run by the lymphocytes. This is the T cells and the B cells. And if you remember back to blood and how blood cells are made, you had a hematopoietic stem cell splitting into a myeloid stem cell and a lymphoblastic stem cell. Now the lymphoblastic stem cells create the T cells and the B cells. So the whole of our specific immunity comes from one class of stem cells in our blood. So they're often called lympho because they act in the lymphatic system and they're found there. So what do we have? Well, we have two parts of our specific immune response. We have cellular immunity, and that exists created by the presence of T cells. T cells are created and trained in your thymus, sitting here just behind the sternum, and so they're called T cells because they're thymus originated cells. And so these check other cells to make sure the cell hasn't been compromised. So this is cell mediated. Now that doesn't cover the whole of our body. We still have fluid in our body, and in particular fluid in our blood, which is the transport system of our body. So if you get into the blood at one point, you can get anywhere else in the body relatively quickly. So we also have a humoral based immune specific system, and humoral means of the blood. So a blood-based immune system, and this is antibody mediated, and these are produced by B cells. And we'll see that in the third of these specific lectures, this being the first. And antibody means it is something that is like a tag you just stick onto the offending person, like a kick me sign, and then everyone else crowds around and gives them a kick. So that's our cellular and our humoral. What we're going to look at here is passive and active immunity and what that means, what are antigens and antigen determinants, and finally what an antigen presenting cell is and how it does what it does. Okay, so passive and active immunity. Well, passive immunity means that you don't have to do anything to get it. So you can get that in two possible ways. You can get natural passive immunity. I've written down here as NP, natural passive, and you get that through your mother when you're developing in the womb through the placenta, and of course when you're first born. That very first breastfeeding gives the baby what we call colostrum, and that is a very, very rich mixture of antibodies, etc., that will sit like sera inside the blood and activate when pathogens come into the baby's system. Now this is very, very important, this first breastfeeding. Even if the person isn't going to breastfeed from that point onwards, they must do the first one because of the importance of the colostrum and the immune response, because a baby doesn't have an active immune system, therefore is very vulnerable to all types of infection. Only what they've got from their mother will keep them going for about the first 18 months of life to up to 2 years whilst the immune system develops. So that's natural passive, breastfeeding, and also through the placenta. But you can have active passive. Now this is where you're given the passive immune system. And this is serum. Sometimes you see in these films where there's a big outbreak of something, and they have to develop the sera in order to save everybody. So the serum, the good example of this is an antivenin, which is raised against snake bites and spider bites and things like that, but in particular snake bites. And antivenins you can inject into someone and they will mop up and interact with the toxin that the snake has injected into your bloodstream, but they won't stay around. They'll get used up and then they'll leave your system. They won't stay there. They'll have no memory. So passive immunity is about having no memory. It's specific, but it won't exist long term. So artificial, I'm thinking we'd say active, which I apologize for. Artificial passive immunity comes from an injection of serum, something like an antivenin for a snake bite. So then we look at the active side. We've got natural active and artificial active. Now, in natural active acquired immunity, you get ill, and your immune system deals with that illness, and eventually you get better after maybe feeling a week to 10 days of feeling pretty rubbish. But during that time, you've formed memory cells, both in your B cells and your T cells, and so that you won't be hit by that again. And later on, we'll show you about the response in the humoral system or blood system to that. If you want to avoid getting ill but have an active immune response to quite important diseases such as measles, for example, you can get a vaccination, and a vaccination gives you artificially, because you haven't done it yourself, artificially active immunity, and this does have memory and it lasts a very long time. So a measles vaccine, for example, will last 40-50 years, and after that you might need a booster to try and up your levels if it's an issue. Don't really have time here to go into vaccinations and the importance of vaccinations. I'll just quickly say that there is absolutely, and never has been, any evidence to suggest that vaccination causes autism in anyone or anything. That was completely made up by a group of people who, for their own reasons, don't like the concept of vaccination, and has caused huge damage, and actually caused serious consequences for people that, because of that information, decided not to vaccinate, and their children then went ahead and became ill with some very, very severe diseases. And let's not forget, vaccinations in general are against diseases that can cause permanent damage and even death. So this was a very unfortunate event. Right, so there's our passive and active immunity. We've got naturally passive in breastfeeding placenta, artificial passive in serum. Then we've got natural active by getting ill, and passive, I keep saying that, and artificial active by getting your vaccine. So these are the different ways that you can actually acquire specific immunity. Now most of it, for most of us really, the main way we get anything is that one. We get ill and then we get over it. One quick point on that, the more between the ages of 5 and 25 that we do get exposed to various different types of bacteria and fungi and viruses, etc., the more our immune system learns. In the same way as you go to school from the age of about 5 or 6 through to 18 or beyond to learn, over time you acquire more and more knowledge. Well, so does your immune system. So trying to remain in a sterile environment between the ages of 5 and 25 isn't necessarily a good thing for your immune system, because it does not learn very much about what it needs to do, and therefore isn't very good at doing it. In exactly the same way as if you take a 5-year-old and you do not teach them how to read or write or do numbers or anything, and at 25 send them out into the world to make a job and make a living for themselves. In this environment, that would be very difficult for that individual, no matter how talented they were naturally. So keep that in mind. Antigen or antigenic determinants. Well, what is an antigen? An antigen is something that sticks out of the surface of the pathogen, whether that be a bacterium, whether it be a virus, a fungi, amoeba, whatever it happens to be. It sticks out on the surface and is recognizable. You can latch onto it. So it's called an antigen because it will cause an immune response in the host once it's placed in there. So antigens can be all different shapes and sizes on the outside. And so I've drawn 7 possible versions of antigen determinants, because that is what will be broken up and presented to the T cell, or the B cell will actually, through an antibody, attach onto, and I'll explain that a little later. So these are all possible ways of triggering the immune response to say, hang on, this cell is not one of us. Let's do something about that. So an antigenic determinant, or AD, is just something that can create an immune response, usually by the action of an antibody that will attach onto it and tag it with a kick me sign to remove it. So that leaves us the final part, the antigen presenting cell. One of the ways these antigens become shown to the specific immune response is, and also shows a link up between, the non-specific and the specific immune response, is through the process of an antigen presenting cell. Now, as the name suggests, the antigen here, the thing that creates an immune response in the host when introduced to the host, is presented to the specific side of the immune response, so they can basically, like looking at like the bouncer being shown the picture saying, okay, this is the person you're looking for, if they arrive, do not let them in, or if they do, in the cells version it would be destroy them, but you wouldn't do that as a bouncer, or if you did, you wouldn't have a job very long. But that's the idea. So I've drawn the basic process, and this is shown in your PowerPoint lecture as well. Excuse me. So you have your bacteria. Now what I didn't do, because I'll have to do it now, is it's got little things sticking on the outside of the bacteria, and these are its antigen determinants. It's the thing that's going to be broken off and used. So the macrophage does what it does. It phagocytizes, engulfs the bacteria, and then releases lysozymes, different versions of enzymes onto it, and breaks up the bacterial wall. So the bacteria has been destroyed now, so that's one bacteria out of the race, which is great. But now the macrophage can do one other thing to actually improve the process greatly. It can take the bits that were sticking out on the bacteria, which can act as antigen determinants, which can stimulate the specific side of the immune response as well. It can take that and stick it onto a protein and put it out on its cell surface. So when the specific immune cell comes along, it can actually physically see it. So that's essentially, you've got the picture of the baddie in your inside pocket, so of course no one can see that. And so when you actually meet the bouncer, you can take the picture out and show them. Just say this is the picture. You can take the picture out and show them the picture. They now know it looks like this. If you see a green-topped pen, you're in trouble, kind of thing. So this is the antigen presenting cell process. Macrophages do this, and B cells also can do this. At this level, we tend to focus more on the macrophage because it's a link-up, which is quite helpful in our understanding. So the bacteria gets taken in, it gets broken down into its parts. Most of that just gets shoved out into the body as waste because it's been destroyed now. But we take these little bumpy things on the outside of the cell, which can actually be recognized as not self, these antigen determinants, and we can attach them onto a specialist protein. Now in antigen presenting cells, or APCs, these are called MHC class 2, so major histocompatibility complexes class 2. So they are only present in macrophages and B cells that do this job, and they bind the antigen determinant, and then they sit in the membrane pointing outwards, so that when the specific immune cell, and it's a cellular based cell, so it's a T cell because this is a cell, so the helper T cell comes up and it has a binding site for the major histocompatibility complex class 2, and it binds on, and then that's when it finds, oh hang on, there's this new antigen determinant sat on this protein. I need to do something about this, and as we'll see when we look at that in the next section, it sends out 3 different signals. One to macrophages to get more active, one to other T cells this is what you're looking for. Hang on again. Here's what you're looking for. When you find this stuck on the outside of a cell, kill it. So that's your cytotoxic Ts. And the same message sent to the B cells in the blood, saying, right, when you find this in the blood, tag it with a kick me sign. Okay? So it does those three things. So the bacteria gets engulfed by the macrophage, broken up. The parts that can be used for recognition are sent out onto specialist proteins and onto the outside of the macrophage, or B cell if it happens to be a B cell. And this is the MHC class 2 with the bacterial or fungal or viral antigen determinant. Helper T cells bind onto that and then can coordinate with the rest of the immune system to speed up and increase the response. So that's the role of an antigen presenting cell on APC. It uses the antigenic determinants on the invading cells against them. This is all I want to say about the introduction to the specific immune response. I know it's complicated. Please, take your time, but get these major points in, because when I do these talks, this is the most important stuff that you need to take away from this section. So thank you very much for listening. When we come back, we'll then look at cellular-based immunity and T cells.